Hello and welcome boys and girls, doctors to 𝗏𝗏𝗃 in which we are discussing general microbiology. This is the 7th class and we are discussing general bacteriology from this class onwards. In this class we will particularly discuss the stains, and in particular I will teach you about the Gram stain, the Acetone stain, some other staining. Then we will look into the classification of Gram positive and Gram negative. So in order to understand the bacteriology, my dear friends, this is a must know thing to be seen. This is a must know video. Okay, so please let us understand first of all Gram staining. So this is a viva question which is very important, and in fact for any examination for that matter, Gram staining is very important. So what you do, first of all, you take a slide, just like I am taking the slide, and I am going to take the sample. For example, I am having a sputum sample. I am going to spread that sample and then heat it. That leads to fixation of the sputum sample with the glass slide. Okay, so this is called as heat fixation. So smear the sample on a slide, then fix it with the heat. That is called as heat fixation. Then I am going to put a stain. Which stain? Can you see this color properly? This is blue color. Okay, is it visible or should I zoom it? Okay, this is blue, yeah. So then you will put a crystal blue or crystal violet, okay, and then I will wash it with water. I'll wash it with the water, okay, and after washing it with water, now I will put some iodine, okay. Now I will put some what? I'll put some iodine on it, okay. So I'm putting the iodine. Again, I will wash it with water and then I will decolorize with the alcohol. Now I can't show you alcohol in this class, huh? Okay? Just remember that we'll wash it with the alcohol, and then I'll put a counter stain which is a red stain. So this red stain I'll put and again wash with water. So wash with water is your choice. Some people do, some don't. But the 5 important steps is, first of all, you will do the heat fixation. Next you will put the crystal violet blue color. After that you will add iodine solution, then alcohol, and in the end the counter stain. So there will be 2 bacteria which will be seen. One will be pink, just like pink, and some will be purple, just like purple, pink, purple. Okay, my dear friends, let's now talk what are the important points, what can be asked. They'll ask you what is the primary stain. So the primary stain is nothing else but the crystal violet stain, the blue color stain. Okay, so this is the primary stain. Next they will ask you about the iodine. Why do you add iodine? So please remember, iodine is responsible for adhesion of CV. What is CV? Crystal violet. Adhesion of crystal violet, right, to the Gram positive bacterial cell wall. Crystal violet, obviously it is going to attach to Gram positive only with the help of what? Iodine. Okay, but that is not iodine, it is Gram iodine, and that is why it is called as mordant. Okay, then you will have to wash it and decolorize with the alcohol. So alcohol, why will you add alcohol? Alcohol is decolorizer. Okay, alcohol is a decolorizer. Just like in your life also, you drink alcohol, all the bad colors of your life will fade away. So alcohol is a decolorizer, and then you will stain it with safranin red. Okay, so Gram positive bacteria, because they have got thick peptidoglycan, it is going to retain the crystal violet and it is going to appear purple, whereas Gram negative having thin peptidoglycan, they do not retain the crystal violet, they will retain safranin, and that's why it is going to be pink in color. Coming on to the other staining techniques. What are the other staining techniques? So remember, sometimes the stain itself remains with the bacteria. For example, positive staining means, just like Gram stain, the stain is taken by the organism. Some are the negative stains. Okay, negative stains means, now people say Gram stain is a negative stain. No beta, Gram negative bacteria is there, but that the stain is not negative. Negative stain means that only the background will be colored, but the organism will not be colored. In Gram staining, organism was colored, but here organism will not be colored, like Cryptococcus, like syphilis. You leave Cryptococcus, but syphilis is actually very important. Syphilis is a very, very important example of negative staining. Also like Indian ink, nigrosin, okay, black, okay, Indian ink, remember it just like it's a black ink. Nigro means black, so nigrosin is again black. Then you have got a simple stain. Simple stain means like methylene blue is a simple stain. Differential staining. Now differential staining means gram stain is going to differentiate gram positive, gram negative. Similarly, acid fast. So acid fast stain is going to give you the acid fast organism, non-acid fast organism. So these are differential stains. There are some special stains. The acid fast stain is modified at different temperatures to form the Zzell-Nielsen stain, and then the Cugnon's stain. So Zzell-Nielsen stain is called as Zn stain. Cugnon's is the cold temperature Zn stain. Okay. Coming on to organisms which are not taking gram stain, mnemonic is scream, or you can say the mnemonic is creams. Okay. So how to remember? Remember anything which is ending with the word ia, like chlamydia, rickettsia, ehrlichia, mycobacteria. They are not going to take the gram stain. So Ria, my friend, oh my god. Anyways, Ria, your friend, does not like gram. Okay, she never likes gram stain. Okay, and then you have got 2-3 more examples: anaplasma, spirochetes, and mycoplasma. This you can remember. So Ria, plasma, these are the things which are not going to stain by the gram stain. There are some special stains also. So remember one stain, I'll tell you a mnemonic to remember. Chan stain, cell wall. So you can easily remember C for Chan, C for cell wall. Fulgin stain. So remember A, B, C, D, E, F. So D and E and F. D for bacterial DNA, and this is the fulgin. Okay. If you have any confusion with this mnemonic, you can remember gen, fulgen, gen for genetics. So genetics means DNA. So fulgen. Then Welch spore. So have you seen spores? Spores are like this, something like well. So well and spores. Silver and Lifsons. So remember flagella has a lot of L's, F, L, A, G, E, double L, A. So L, L, L. So silver impergonation and Lifsons, both of them have got L. So flagella is by the silver impergonation and Lifsons method. Then you have got Muir stain. Now what is Muir stain? Remember like this, I have a cap and my cap has a symbol of Indian flag. So my cap, India's flag. So what I am trying to tell, cap means capsule. So capsule. So in order to stain capsule, you have got my, that means Muir stain, and India's flag, that means Indian ink. So Indian ink and Muir stain, both of them are there. Then very, very important thing, very, very important. I have already told you when we were discussing the storage granules, the plasmids, I have told you that storage granules are there as a protective mechanisms which are being not protective, but you can say as a preparatory thing possessed by the diphtheria. Okay. In order to stain those storage granules, we have got 3 stains. One is Albert stain, Ponder stain, and Neisser stain. Okay. So how will you remember? Albert, my friend Albert, he ponders storage material in Nissan car. So we have a car, Nissan is a brand, Nissan car. So Albert, what does Albert do? Albert ponders, he keeps storage granules or the storage material where? In the Nissan car. So Albert, Ponders and Neisser, these are the ones for the storage. For the storage, Nissan car, C for Corynebacterium diphtheria. Okay. Fluorescent stain, the ones which show fluorescence. What are these? Auramine, rhodamine, Mycobacterium tuberculosis. Auramine, rhodamine, M for Mycobacterium tuberculosis. Very easy and simple to remember. Auramine, rhodamine. Then chalcofluor white. So chalcofluor, fluor, F-L-U-O-R, F for fungus and F for fluorescent also. So fluorescent stain for fungus is chalcofluor white. Next you have got acridine orange. So orange flavor. So there is an Odomos which is of orange flavor. Okay. So remember orange flavor Odomos, and Odomos is for what? For malaria, Plasmodium. So acridine orange is for Plasmodium. Okay. Now coming on to acid fast staining. So what are the different organisms which are picked up by the acid fast stain? So acid fast are obviously all the mycobacterias. All the mycobacterias, be it your tuberculosis, Mycobacterium tuberculosis, atypical mycobacteria and Mycobacterium leprae. Now, a lot of students ask me, sir, is it important to remember the concentration of H2SO4? Yes, my dear friends, H2SO4 is the decolorizer, and depending upon how much H2SO4 is being utilized by bacteria, or I mean the slide to get decolorized, we can tell which organism is this. If it is getting decolorized by 5%, it is leprae. Okay, I remember like this, L for low H2SO4 is required, low H2SO4 is required, right, leprae. And if it is atypical mycobacteria and Mycobacterium tuberculosis, they want 25%, okay. So remember like this, atypical has T, this is also going to have, TB is going to have T, so TT means 25, okay. What are the other organisms? Nocardia rhodococcus, very important. Can you see I have written no here? No. So this is a rod also which is formed. So rhodococcus and nocardia, both of them also utilizes acid fast staining. Mycobacterium leprae, L for leprae, L for legionella. So both of them are going to use acid fast stain. Then you have to take care of these parasites. Take care, parasites. Why? Because you will get identified by acid fast stain. So which parasites should take care? T and C. T for Taenia solium X and C for coccidian parasites. Taenia solium and coccidian parasites, these are parasites. Next is bacterial spores. So the spores of bacteria, okay, they just require slight H2SO4. Slight H2SO4 means they just require 0.25% H2SO4, okay. Now coming on to gram-negative bacteria and gram-positive bacteria. So whenever you want to understand about the gram-negative and gram-positive bacteria, first of all you have to understand and keep one thing in mind, that the tables which I am going to show you, the charts which I am going to show you are of vital importance. They are very important. Why? Because you know what? Whenever you will write the laboratory diagnosis, okay, first of all let me just share you the trick to write the laboratory diagnosis. So whenever you are supposed to write the laboratory diagnosis, the very first thing which you will write is the specimen collection. Like for example, if it is TB, you will collect the sputum sample, right? Next you will write the stain, okay, the stains which we are going to do, and obviously a diagram of stain is important. Then after staining you will write culture. So culture, specific culture media, and obviously culture method. This we will discuss in our next video. So specific culture media, specific culture method, some transport media if it is there, some differential media is there, right, like that. Then after culture you will come to antimicrobial susceptibility testing, that we will discuss in next class. Then you will come on to CBNAT or TrueNAT. Next, sixth one is ELISA, PCR. Then some biochemical tests which we will discuss in the most probably in the second class. Okay. So all these things are important. Fine. Now gram-negative bacteria, if you see the classification, gram stain is having gram-negative bacteria. Gram-negative bacteria has cocci, rods, coccoids, okay, and 𝗵𝗵𝗶𝗵𝗵. So these are the four shapes which are there in gram-negative bacteria: cocci, coccoid, rods, and 𝗵𝗵𝗶𝗵𝗵. So in cocci you have got maltose fermenter and maltose non-fermenter. So maltose fermenter, M for maltose, M for meningitis. So Neisseria meningitis is the organism which is gram-negative, and cocci and you have got maltose fermenters. And non-fermenters is gonorrhea, Neisseria gonorrhea. So basically in cocci you have Neisseria. The Neisseria which is maltose fermenter is Neisseria meningitidis, and which is non-fermenter is gonococcus. So no, I am not going to get fermented, that is non-fermenter. Next you have coccoid. In coccoid, the examples are He and Ella. So cocoid, example are he and ella. Ella means Brucella, Pasteurella, Bordetella, and he means 𝗵𝗵𝗵𝗵 influenza. These are cocoid. Next is rods. So rods either will have lactose fermenters or lactose non-fermenters. So lactose fermenters are fast. So you can remember 1 second. 1 is 1 in Hindi. 1 second. So 1 means what? Lactose fermenter, fast fermenter, slow fermenter. 1 second is the mnemonic for fermenters. So 1 means E and K. So E. coli, Enterobacter and Klebsiella. Mind you, this is not Enterococcus. I am not writing Enterococcus. I am writing Enterobacter. Because coccus has to be cocci. No, why? How will it be rods? We are discussing rods. Okay. So that's why it is Enterobacter. E. coli, Klebsiella. These are fast fermenter. Mnemonic is 1 second. So slow mein 𝗲𝗃. S and S. Second. So 1 second is the mnemonic for lactose fermenter. 1 means Klebsiella, E. coli, Enterobacter. Second means 𝗲𝗃, which is there, which is a slow fermenter. Coming on to lactose non-fermenter. Coming on to lactose non-fermenters, you have got oxidase negative, oxidase positive. For example, if I talk for pseudo, so pseudomonas, if I talk for O for oxidase and pseudo may P for positive, so it is positive oxidase. If I talk for oxidase negative, the remaining ones Shigella, Salmonella and Proteus, right? And coming on to oxidase positive 𝗵𝗵𝗵𝗵. So 𝗵𝗵𝗵𝗵𝗵 is Vibrio cholera and Campylobacter. Okay. So these are oxidase positive. How to remember? Please see Vibrio. Vibrio is having this O, that means it is oxidase positive. Okay. And Campylo. So sorry. Campylo, O for oxidative positive. Okay. So some more gram-negative diplococci are Neisseria and Moraxella, right? Just remember M and N, diplococci. Gram-negative bacilli, which are anaerobic, are bacterioids. Okay, they are bacterioids. Coming on to the last chart, this chart is for gram-positive. Remember again, you have got cocci, you have got bacilli and some branching organisms. So branching, very easy to remember, A and B coming together, B for branching, A for actinomycetes and NO is nocardia. Okay, clear? Coming on to rods, again easy to remember. Remember, you have got Listeria, anthrax and Clostridium. So how to remember? Remember like this. You have got Bacillus anthracis. Bacillus itself means rod-shaped. So anthrax has to be rod-shaped. Easy to remember. Then you have got only 2 more, that is Corynebacterium, Listeria and Clostridium. 3 more. So how to remember? See the spelling. B for Bacillus anthracis, C for Corynebacterium and Clostridium, L for Listeria. So this is about the rods. Coming on to cocci. So cocci means what? Catalase positive cocci, catalase negative cocci. So catalase positive cocci is the one which likes to stay in a grape-like cluster. So grape I have already discussed. Staphylo has got philo. Philo means love and I love grapes. First thing. Second concept, please build along with me that grapes G and C go together because they are similar and C is catalase positive. C is catalase positive. So catalase positive Staphylococcus in cluster, okay, and catalase negative is going to be Streptococcus. But Streptococcus has a unique property of hemolysis. Streptococcus hemolysis is very common. So you have got 3 categories: alpha category hemolysis, beta category hemolysis, gamma category hemolysis. Remember alpha, पर्व, me, I am alpha male भाई. खुद की तारीफ खुद करने में कितना मजा आता है. How happy you become when you praise you yourself. So alpha. So alpha means P and V. So P for pneumonia, Streptococcus pneumoniae, and V for viridans. Even if you forget, remember viridans. Veer. Veer is a strong person, brave person. Veer is in Hindi. In English we call them as brave person. They are alpha males only. Okay. So pneumonia and viridans are alpha. Beta has got 2 groups, group A and group B. Group A and group B. So, the examples are Pyogenes and agalacti. Streptococcus pyogenes, Streptococcus agalacti. Please remember A for B. So, group B is having A. So, group B, agalacti, Pyogenes is group A. Gamma is going to have Enterococcus and non-Enterococcus. So, Enterococcus is Enterococcus faecalis, non-Enterococcus is Streptococcus bovis. So, is word, if it is coming in the end, it is group gamma, okay, faecalis bovis. So, I hope that just the way I have taught you stains, I just wish that you will also be stained in the success, in the love for my subject. My comment section is always open for you, for your love, blessings, care, support, happy comments, positive vibes, anything troubling you, you can share, okay. So thank you so much, and please don't forget to put your doubts in the comment section and keep on revising. Happy learning. Thank you.